Hi y'all, these are the protein notes. So what is a protein? A protein is a macromolecule made up of nitrogen, carbon, hydrogen, and oxygen. So referring back to our functional groups chart, you should be able to look at this protein structure, which is a polymer in this case, of 7 amino acids. And you should be able to determine what are the functional groups found in these 7 amino acids. So hopefully you were able to find some nitrogens. Here, here, and here, 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 every time. So along the chain. The one that's up here is actually going to be part of its functional group. So, or the R group, excuse me. So the nitrogens here, here, are all going to be playing a role, and we call that the amino group. Next, you should see some classic signs of a double bonded carbon to oxygen, and if it's on its own, you would be able to look at it as a carboxylic acid or carboxylic acid. So right off the bat, we have our stems for our word amino acid, amino group, and carboxylic acid group. Okay, so if you need to refer back to that functional groups table, please do so. Proteins are polymers. Polymers are going to be a larger molecule made of several smaller molecules called monomers. Mono is a prefix that means one. So if I have these amino acids, all of them are considered monomers, and I polymerize them by using dehydration synthesis. Dehydration synthesis would bond each one of these together with one bond, one water lost. So I have 1, 2, 3, 4, 5, 6, 7, 8 amino acids bonded together in this protein polymer. So that means I have 7 waters lost as a reminder. So our monomer is an amino acid. We have this little meme here. What do you call an acid with an attitude? Give me your lunch. An amino acid. All right. So amino acids are building blocks or monomers of proteins. They're comprised of nitrogens, carbons, hydrogens, and oxygens. They have 3 important functional groups: amino groups, carboxylic acid groups, and the R group. The R group is called the R group or R side chain, and each R group is different in every type of amino acid. So these 2 parts are the same every time. In an amino acid, we have an amino group and a carboxylic acid group. The only difference is this R section. Okay, so I like to think about it as different mods or different modifications of each amino acid, and there are 20 different types. 20 different amino acid R group variations. So you can see this amino acid chain or this protein of 1, 2, 3, as if I was going to put them all together. I have my amino group or amine group here in pink. I have my carboxylic acid group in green. And then I have my R group, which is different for each of these amino acids. So tyrosine is the side chain or the R group on this amino acid. So the whole thing is called tyrosine, the whole thing is glycine, and the whole thing is methionine. So we have many different possible combinations. If you think about it, there are 20 different amino acids. And so if we put them together in a way to count all the different types of amino acid combinations, that's a 20 factorial. So if you plug that in, 20 factorial, you would get this whopping number of, here we go, 243,290,2008. 1766. 40000. That is how many different combos there are of the 20 amino acids you could have in the body. Now, you don't have to write this number down. I am just telling you that there are many combinations of these modifications you could have of these amino acid chains and all the different variations. So here's what I'm talking about. Here is we call it a polypeptide chain because the bonds between amino acids are called peptide bonds. So polypeptide means many amino acids strung together with peptide bonds. Here are the different amino acids. If you count them, there are 20 different amino acids here. And strung together with the peptide bonds between them. So they're put together like we already established with dehydration synthesis. To bond together, I need to remove a water. So you would take a hydroxyl group on one and a hydrogen on the other, and you would remove that. Okay, so if I remove that, okay, I get a water lost, and then those bonds connect. Here, see? And the bond that forms between them is called the peptide bond. That's why this chain was called a polypeptide chain. Okay, so now there's more to the story here. So the function of the protein depends on its shape. So here is a shape of a protein. It's its most basic shape called primary structure. This is the unique sequence of amino acids in a protein chain. So this is determined by your genetic code, okay? And it dictates how the protein will fold into its higher levels of structure. A single change in the amino acids can affect the protein's function. So we have not talked about mutations yet, but we will. Now, as you can see, there is a secondary structure. So we have an alpha helix and a beta plated sheet. So this refers to the folding patterns within the protein. So those fold mainly into these two shapes, the alpha helix or the beta plated sheet. So the shapes are stabilized by those hydrogen bonds. You can see the weak bonds here between the oxygen of one amino acid and the hydrogen of the other. And so we think about this like the initial bends and twists in a ribbon that begins to fold. Eventually they fold into a tertiary structure. Tertiary structure is going to be the three-dimensional shape of a protein. All right, and it's formed by the interactions between the various side chain groups or the R groups. So they can include hydrogen bonds, ionic bonds, some that are hydrophobic, so they're afraid of water, so they fold in a certain way, and disulfide bridges, but you don't really need to know that. And quite frankly, the last part is the quaternary structure. Wow, that is when the overall structure involves the assembly of multiple protein subunits into a larger complex. So think of this like a larger protein. Think like hemoglobin, which is a protein that carries oxygen in the blood. It has 4 subunits. So like you would see the 4 different subunits in this structure like this. Okay, for example. So the quaternary structure enhances the protein's functionality. It allows for cooperative behavior between subunits, and the binding of one can affect the others. So it really depends on the function of the protein, depending on how complex its shape is going to be. Okay, so that's going to be your takeaway. The takeaway here is that the sequence of the amino acids and the shapes will dictate the interactions and vice versa, and that helps dictate what the function of the protein in the body. So what do you think would happen if the primary structure, which is this first part of order of the amino acids, change? Well, that could lead to a very significant mutation. For example, we have hemoglobin here. Remember, hemoglobin was our special protein that has the 4 subunits in the blood that helps carry oxygen. Now, if you have a substitution for a amino acid that is not a good one, it's called sickle cell as one example. Sickle cell is an inherited trait in African and Middle Eastern countries, and it is a deformed red blood cell. And not all red blood cells are this sickle shape, but just, many of them. And that reduces the ability for that person to carry a lot of oxygen. But it actually is a selective advantage against malaria, which is a virus that spreads via airborne mosquitoes or parasites. So more on that when we talk about genetics. But let's move on. Other functions of proteins, pretty much everything. They support the animal, so structurally, like keratin, hair, skin, nails, horns, shells, all those things are made of keratin. Collagen, so your bones, your tendons, your ligaments, all of those are made of protein. We also have special proteins that are helping us in terms of little molecules that help speed up reactions in the body. We call enzymes. So special enzymes are, we call those catalysts. They speed up reactions. That's why we have this little meow, because they're catalysts. So anyways, they speed up reactions. So sucrase is a special enzyme that breaks down sucrose into its monomers. Lipase breaks down lipids. Let's go ahead and change that to lipids. And flipase flips phospholipids in the cell membrane, because maybe, they got to be flipped for a certain reason. So here's how enzymes work. We're going to talk more about this later, but basically they fit in with their molecules and they help break them down quicker. So other functions, transport. So of nutrients across the membrane, for example, or through the body. So they can serve as channel proteins across a cell membrane, which is the outside part of your cell that helps. It's like basically a tunnel for molecules to cross over. Those proteins, those channel proteins, are made of amino acids. And then we have hemoglobin. That was our example from today of a more complex protein that carries oxygen throughout the body and the bloodstream, and loads up oxygen and unloads carbon dioxide. We also have proteins that serve as immune function, so a defense against foreign invaders, and they help protect the body. They are created and rejuvenated over the years. Sometimes you need periodic exposure to certain viruses or pathogens to maintain the antibodies. Sometimes you might need a vaccine in order to do that without getting an actual infection. So we'll talk more about this stuff primarily in my anatomy class, so you should stay tuned. Hormones are another example of some proteins. So they help send signals to cells and organs to do certain things. They're chemical messengers, such as insulin, to take in glucose from the blood and help use that for cellular functions. It says, why are proteins able to have so many diverse functions? Well, we have 20 different amino acid R groups. So we have that big old number that I already gave you on that previous slide of all the different combinations of the amino acids that we could have to make proteins to do all the things that we need to do in the body. So where can you find proteins in terms of in the body or consumption? So they are made in the cells, and they are primarily found in hair, skin, nails, bone, muscles. You might find them everywhere. Okay, in foods we eat, there would be muscle. So muscle on animals is considered meat. Eggs, nuts, legumes, etc. So how would I test for proteins in the laboratory if the protein solution is clear and I can't taste it? Well, you could test it with this indicator solution called Biuret's indicator. It's spelled like this. It is a light blue normally without, mixing with anything. And what you do is you mix it with a solution, and if it stays light blue, this is a negative test for proteins, which means there's no proteins there. And on the flip side here, that means proteins are present if the light blue color turns into a light lavender color in the presence of a protein. Okay, so now you're going to go ahead and click this link and complete the checkout for me. Have a great day.